Hi everyone, welcome back to A View On, Lonza's podcast on the latest science and trends in pharmaceutical manufacturing. Today, we're exploring how process analytical technologies, or PAT, are transforming biologics production from the lab bench up to the full-scale manufacturing. By bringing advanced sensors and real-time analytics into every step of the process, PAT enables teams to see what's happening inside their bioreactors, reduce variability, and make data-driven decisions faster than ever before.
Having that process knowledge is so important for our customers. And if we can do something on this journey to give them better insights into their process, and then together come up with savings, either by, reduced media consumption or, improved process control, it really gives them an advantage in the long run.
We spoke with 3 experts from Lonza's PAT Center of Excellence to understand how this unique set of technologies is being implemented across the industry. Kerry Mason, Thad Webster, and Ryan McDougall are at the forefront of deploying these technologies, which benefit our customers' projects and timelines, and ultimately their patients. From upstream cell culture to downstream purification, PAT is redefining process control and how the industry ensures quality, consistency, and speed. This is A View On, a podcast brought to you by Lonza. Hi, Kerry. Thanks a lot for joining us. So, as a head of the PAT Center of Excellence at Lonza Portsmouth, could you explain to us what exactly we mean by process analytical technology?
Process analytical technology, we kind of like to say it's pointing light or a flashlight into the dark corners of your process and getting information, right? So we're trying to measure things that historically haven't been measured or were measured with offline technologies that results in delays or taking time to get the answers you're looking for.
And what kind of challenges do traditional biomanufacturing methods face today?
So traditional biomanufacturing methods right now are very recipe based, and we use a lot of standard tools that we've used for years. So what that has really led to is a challenge when it comes to wanting to do something better or faster, or maybe be able to answer our requirements for sustainability.
So, how does PAT fit into that picture? Where can it make the biggest difference?
So PAT can be used in a development lab where we can actually speed decision timelines. So instead of taking your samples and sending them to a dedicated lab, you can now start to get that information real time and make faster decisions when you want to do the next iteration of your study. PAT can also really work wonders when we're putting it into our operations and manufacturing because it can start driving reproducibility in our processes. So we can start looking at ways to reduce the variability in our processes and then eventually to enable us to go to intensified manufacturing or continuous manufacturing.
So, would you say that a continuous manufacturing approach is an industry trend? Is that transformation happening yet?
The continuous manufacturing approach has its needs, so it really can answer a lot of problems when you're dealing with potentially molecules that have stability issues or small volume footprints and you need high capacity. Those processes are out there. I think our approach that we currently use with our batch processing is still the expectation in our industry right now.
Yeah, that sounds like quite a bottleneck. Are there any other challenges slowing down the progression in implementing PAT more broadly?
So the challenges associated with implementing PAT is we are in a very highly regulated environment. It's very hard to make improvements upon things that we've already improved upon for a very long time, right? So trying to find that business case for what makes this a little bit better than the other way of doing it, right? Because we are very good at what we do. We release lots all the time, golden batches, diamond batches. We're in control.
Oh, I like that term, diamond batch. What does that mean?
Absolutely no deviations and on-time release.
That's definitely something to strive for. So now I want to take a step back. How does PAT help bring more transparency into upstream processes, like understanding what's happening inside the bioreactor?
So, yes, in a way, we have implemented some monitoring of our bioreactor health for a very long time with tools like, uh, you know, cell viability, pH, and things like that. But when it comes to looking at the complex conditions that our cells need to be in, maybe monitoring metabolites and monitoring consumption of glucose and things like that. We haven't really gone there, right? What PAT enables us to do is to start getting more insight and being able to enable us to make better choices and more informed choices for how to optimize our processes.
When a PAT is applied successfully, what benefits are already visible today?
So right now we're actually seeing that where we have rolled out PAT in our bioreactor systems, which are as limited in scope for all of our biologics network, but where we have rolled it out, there has been increased customer satisfaction with knowing that we have additional monitoring that gives us this insight. And then also our MSAT colleagues have taken this technology and have used it, in order to make process decisions in order to enhance our process robustness. And then also it was used at one point to intervene in a controlled manner before a processing batch went out of spec. So they're actually able to use that in order to preemptively control our processes in order to prevent batch losses, which is absolutely amazing.
That's quite a powerful outcome, both for process control and customers, right? When you talk about bringing more visibility into a process, what kind of analytical tools are we really talking about? For instance, methods like Raman or IR, so infrared spectroscopy, that can measure what's happening inside the bioreactor in real time.
So, everybody has the ability to do ramen. Lots of people can use ramen. I think where Lonza is at an advanced point is where our offering is, and the fact that we offer a platform way of using ramen and our ability to reach out and do this. One of the things that, recently has come up is who's driving PAT? Is it the individual companies with their one molecule, their one pipeline, or is it really the CDMOs? And I think the CDMOs are playing a very big role in this because of the fact that we can reach out to so many customers, so many molecules. And, for me, when I had conversations with some of the people that happen to also be our customers, they were very pleased to know that we are getting real-time information. Because one of the big things in industry right now is real-time release, accelerated release, getting that additional critical information. Having that process knowledge is so important for our customers. And if we can do something on this journey to give them better insights into their process and then together come up with savings either by, reduced media consumption or, improve process control, it really gives them an advantage in the long run that we can offer them. Whereas some of our competitors may not be as far on the journey as we are.
What are some of the other techniques emerging as part of PAT?
So one of the tools that we actually have really developed as well is a miniaturized HPLC system that does titer.
Wait, I'm picturing a tiny HPLC now. And for anyone less familiar, that's the lab instrument that is used to measure how much product you have at a given step, right? So, how small are we talking?
Honestly, it looks like a shoebox. Looks like a shoebox. And so this is very critical because in the past, when we got to a point in our processes, so our large scale facilities run 24/7, right? And so when they get to the harvest titer step, they traditionally send a sample to QC. That QC will then do a traditional HPLC method in order to get the titer value. So what's amazing with this is that it uses a miniaturized HPLC. So you can put it on the manufacturing bench. No sample cleanup, no nothing besides a point true filter. Take condition clarified harvest, filter it, put it into the machine, and within 8 minutes, you have your titer value. Instead of taking the sample to QC, QC running in triplicate, QC then coming back with a result, and then you being able to then go.
Oh, that's super exciting. Thank you, Carrie.
Thank you.
Carrie Mason just painted a clear picture of how PAT empowers teams with deeper insights into their processes and helps them act faster, improve process optimization, and prevent losses even before they happen. These technologies are already showing real results from Raman spectroscopy to miniaturized HPLC systems. Next, we turn to Thad Webster, who brings a closer look at how these tools are implemented across modalities, from mammalian cell culture down to microbial and even mRNA systems. Thad, thank you for being here. So, as the person developing the next generation of at-line PAT tools, could you start by telling us how your team applies PAT across different biomanufacturing processes?
We specialize in evaluating and implementing the latest PAT tools that can help Lonza not only measure but also control its processes better. Our typical application is monoclonal antibodies, but the tools we develop could be applied to any bispecific, any fusion protein, anything that we might produce within our integrated biologics facilities.
And do you see these tools transferring easily across other modalities?
I think Lonza has positioned itself really well with the PAT tools we developed today. We started off in mammalian because that's where most of our focus was, but the tools that we use could also apply to microbial or mRNA depending on the application as needed.
We also just heard Kerry talk about that miniaturized HPLC system for fast titer checks. How does your team take those kind of tools further into everyday operations?
The team I'm in focuses on both upstream and downstream. So I now head up what we call the outline PAT team. So we are looking for tools that would typically be used in the QC realm, and those are perfectly fine. They give us the answers we want. The downside is they tend to take a very long time to get the answer. So if I'm a development scientist or if I'm a person on the floor and I'm waiting for a titer measurement to forward process to downstream and it takes 8 hours to get, that's 8 hours of sweet time. That's 8 hours of development time that's gone. So if I can find a tool that gives me the same answer that the QC release test would give me, but I can put it in the hands of the operators and they get that answer in 10 minutes, we've now created a throughput efficiency that we previously didn't have. My group is looking at ways to empower the people who are actually going to be pulling the samples to get the information they need and maybe not rely on QC unless it's absolutely necessary for a release test.
Oh, that really speaks to how PAT empowers people on the floor to make faster decisions, right? But looking ahead, what do you see as the next big step in PAT implementation? Where is this evolution taking us towards?
But the beautiful thing about process analytical technologies is that they're constantly evolving and we're not trying to reinvent the wheel. We're building on what was already a QC gold standard and we're finding ways to make that work for people who may not be QC trained scientists. And the goal is not to remove the QC scientists from the picture completely. It's to have them only do the tests that are absolutely required for release while freeing up operations to forward process as needed. For upstream, it's about removing the need for offline sampling completely. So right now with Raman, we've tackled a few of the things that we want to either measure to control or measure to trend. If we could find ways to use Raman or other tools that could get rid of all the offline sampling completely, so the operator doesn't need to do that anymore, except by exception, then you'd see, in my mind, a huge benefit to Lonza because now we're having the operators monitor the process, trend the process.
How much does broader adoption depend on guidance from health authorities? Is the implementation of PAT also a regulatory decision, in other words?
That would certainly facilitate the adoption, I think, not only within Lonza, but within the broader industry.
Yeah.
The regulatory bodies have said that they do want to see process analytical technologies rolled out. They think that they provide more value because we are now seeing what we're doing. We're measuring more. We're able to control better. So all those things lead to a more consistent product that is safe and efficacious for the patients that we're going to give it to at the end of the day.
Okay, amazing. Thanks, that.
Absolutely. My pleasure.
And finally, we turn to Ryan McDougall, who's applying PAT to downstream processing, where rapid, real-time analytics can transform how purification steps are monitored and controlled. Ryan, when I think about in-process analytics, first thing that pops into my mind is a bioreactor, where you can control in real time how happy the cells are you're growing. And, when it comes to the levels of oxygen, sugar levels, pH, etc. But what does PAT look like when you move to downstream processing, when you're already purifying your protein of interest or molecule of interest?
Great question. This looks like our initial target for inline PAT in downstream is geared towards at least being able to monitor where the product is and being able to quantitate how much product there is. In the downstream space, obviously, we're not looking to make cells happy. Hopefully by that point, we've removed all the cells and aren't introducing any new ones. But we're really needing to be able to track the product. Our initial forays and initial implementation is geared towards being able to track that in real time and wherever possible, eliminating the need for offline sampling, whether that's in the manufacturing area, in the suites, or submission of samples to QC. As we're continuing to explore, obviously, some of these technologies have the ability to not just give you the product concentration, but could also potentially give you an idea on product quality as well, whether that's aggregation, whether that's for when we're looking at bispecific formats, being able to differentiate between the correctly paired heterodimer compared to homodimers that you don't want in the process and would like to be able to remove.
And what's the biggest driver behind eliminating offline sampling?
So the less time that you're taking between steps to analyze and confirm that things ran as the process intended, the more opportunities there are going to be to keep that process moving forward, reduce suite time, and ultimately lead to being able to increase the throughput in the manufacturing suites.
Ryan, in your view, is the applicability limited to proteins?
Process analytical technology, PAT, is an enabler. So it would have applicability whether you're purifying proteins, whether you're growing cells in a bioreactor, whether you're taking that purified protein and conjugating some sort of active toxic substance to it, or whether you're refolding a microbial protein, or whether you're refolding or transcribing and creating an mRNA vaccine.
And looking ahead, where do you see the biggest opportunity for innovation in PAT?
There's always room for improvement. In upstream, there's been a lot of benefit demonstrated already in terms of being able to utilize Raman spectroscopy and help support and drive towards having a process that is more optimized in terms of feeding strategy. The challenge is trying to leverage those same opportunities in the downstream space. There are challenges associated with downstream related to just the process time. The whole cycle is running for maybe several hours, but the critical step where the product is being eluted is running in the order of minutes. So the need for having not only correct information, but that correct information very quickly presents unique challenges in the downstream space. So there's still plenty of opportunities there where taking the measurements that we are and being able to translate those measurements into actionable information that's accurate and enabling those correct decisions to be made. And I believe that's where it's possible that, moving beyond just traditional modeling and moving into more so maybe machine learning, and they become more of an enabler to bring some of the spectroscopic methods into downstream.
Amazing. Thank you for your time and insights, Ryan.
Thank you very much.
Thank you for listening to A View on PAT in Pharma Manufacturing. From upstream monitoring to real-time analytics in downstream processing, process analytical technology is reshaping how pharmaceutical manufacturing operates by providing deeper visibility, faster decision-making, and greater process control. Carrie, Ted, and Ryan from Lonza's PAT Center of Excellence have shown us that these new technologies enable a real shift towards data-driven, agile manufacturing that can ultimately accelerate therapies to patients. Join us next time as we discuss emerging trends in the early development of pharmaceuticals. If you cannot wait, head over to lonza.com/a-view-on to listen to our previous episodes, subscribe to never miss an episode, and access additional materials and interviewee info. Bye for now.